So picture this. You open the refrigerator door, right? And, middle shelf, maybe pushed slightly to the back. There's this plastic-wrapped chicken breast that you bought, I don't know, a few days ago, or maybe it's a steak.
Yeah, we've all been there.
Right. You pull it out, and immediately your brain just starts running this, primal, incredibly complex algorithm.
It really does. It's an instinct.
Yeah. You look at the color, you peel back the film a little bit and take a hesitant sniff.
The dreaded sniff test.
Exactly, the sniff test. And maybe you even poke it to see if it feels, slick or weird.
Right, checking the texture.
And in that, 10-second window, you are essentially trying to play forensic pathologist to a piece of muscle tissue, right? You're trying to figure out if it's still dinner or if it's, crossed this invisible threshold into becoming a literal biological hazard.
It is, it's a really profound transition when you think about it.
How so?
Well, I mean, we buy a chair, right? And it remains a chair. We buy a car, and it mostly stays a car until the parts wear down over, years.
Right.
But meat is fundamentally different. It's a biological landscape that is in a constant state of, and this sounds dramatic, but rapid, violent decay. From the very second it is processed.
Wow. Violent decay.
Yeah. So when you stare at that chicken breast in your fridge, you are looking at a ticking biological clock.
I always used to think of meat as just a passive object, like a carrot, A carrot might eventually just dry out and get rubbery.
Right, it desiccates.
Yeah, it just kind of gets sad. But meat, it doesn't just get sad, it becomes a war zone.
A literal war zone.
And so for today's deep dive, I really want to figure out the underlying physics and chemistry of that war zone. Why is this specific type of food such a massive, irresistible magnet for microbial life?
Well, to really understand that, you have to kind of completely discard the idea that meat is food for humans.
Okay.
From an evolutionary perspective, human consumption is completely irrelevant.
Right. Nature doesn't care about our dinner plans.
Exactly. You need to look at that steak for the perspective of a single bacterial cell.
Okay, I'm a bacterium.
Right. And to a microbe, raw muscle tissue is, it's basically the most perfectly engineered culture medium on the planet.
Really? The absolute best.
Without a doubt. It checks every single box required for explosive, exponential cellular replication.
Okay, I'm trying to picture this from the microbes' perspective. They need water, obviously. Everything alive needs water. And meat is, roughly 75% water?
Roughly, yeah.
Yeah.
But it's not just the sheer presence of water. It's the availability of that water.
Availability? What does that mean?
In food science, we call it water activity.
Okay.
The moisture in fresh meat isn't locked away in, indigestible structures or bound up tightly. It's highly accessible.
So they can just drink it right up?
Basically, yes. It allows microbes to transport nutrients across their cell membranes with minimal energy expenditure. It's easy access.
Gotcha. So water is the baseline.
Right. But water is just the baseline. The real draw, the thing they really love, is the nitrogen.
Nitrogen. Because, wait, nitrogen is for protein synthesis, right?
Precisely. Microbes need nitrogen to build their own amino acids, to build their own DNA, their own cellular machinery.
So they're stealing the animal's building blocks to build more of themselves.
Exactly. And meat is profoundly rich in highly available nitrogenous compounds. It is a dense, concentrated stockpile of the exact building blocks a bacterium needs to clone itself billions of times over.
It's like dropping them into a Lego store where all the pieces are exactly the ones they need.
Perfect analogy.
Yeah.
And then you add to that a really rich supply of minerals like iron, zinc, magnesium, and all these accessory growth factors like vitamins.
Oh, wow.
Yes, you have this environment that actively supercharges microbial metabolism.
Okay, so they have the water to move things around. They have the nitrogen to build their cellular walls and their DNA, but they still need fuel, right? They need energy to actually run all this machinery. Now, I know meat contains some stored sugars, specifically glycogen, right? I'm assuming that plays a big role.
It plays a monumental role, but perhaps not in the way you might think.
Oh.
So glycogen is an animal's energy reserve. It's this complex branched chain of glucose.
Like animal starch.
Exactly. When bacteria land on this tissue, they encounter these glycogen reserves and they begin fermenting them.
Fermenting, not respiration, but fermentation.
Right. Fermentation is this rapid, highly efficient pathway for microbes to extract energy without needing oxygen.
Okay, so they can just burn through it super fast.
Yeah, it allows them to fuel their metabolic processes incredibly quickly.
So they hit the glycogen, they ferment it, and their population just explodes.
Yes. But The fermentation of that glycogen by the bacteria, and actually even by the meat's own dying cells, produces something else as a byproduct.
What's that?
Lactic acid.
Ah.
And this brings us to the final and really most critical environmental factor, the pH level.
The acidity.
Right, correct. Living muscle tissue has a fairly neutral pH, usually around 7.2.
Okay.
But after death, as that glycogen breaks down into lactic acid, the pH starts to drop.
Because acid is being produced.
Right. Ideally, it drops to about 5.5.
Okay, 5.5, is that good? Bad?
Well, a pH of 5.5 is slightly acidic, which is actually somewhat hostile to a lot of bacteria. It's essentially nature's built-in preservation method.
Oh, that's wild. So the muscle tries to preserve itself?
In a way, yes. However, compared to something highly acidic like a lemon, which is down around a pH of 2,
Right.
a pH of 5.5 to 6.0 is still well within the Goldilocks zone for a massive variety of spoilage microorganisms.
So it's not acidic enough to stop them completely.
Exactly. It's still a highly favorable pH for their survival and proliferation.
Okay, so spoilage isn't just a side effect then. It's like the inevitable biological consequence of putting a perfectly balanced microbial food source in a warm room.
That is the crux of it.
The microbes just show up, they consume the moisture, the nitrogen, the glycogen, and in doing so, they excrete waste.
Yes. And here's the thing. The bacteria themselves might not even be inherently pathogenic.
Wait, really? Like they wouldn't make you sick?
If you consumed a lot of these spoilage bacteria in pure isolation, many of them wouldn't necessarily cause an infection.
Wow. Okay.
But their metabolic processes, their digestion of the meat, produce massive amounts of chemical byproducts, acids, gases, volatile organic compounds.
It's the waste that's the problem.
Exactly.
Yeah.
That accumulation of microbial waste is what we define as spoilage. It's what makes the food smell foul, get structurally mushy, and eventually become toxic to us.
Okay, which means the battle against spoilage isn't a question of if. Like, we aren't trying to prevent it indefinitely. We are just trying to delay a mathematical certainty.
That's a great way to put it. We're just buying time.
But, okay, here is the mechanism from the sources that absolutely threw me.
Let's hear it.
I always just assumed the spoilage clock started the second. Like, a rogue bacterium drifted down from the kitchen ceiling and landed on the steak.
A lot of people think that.
Right, but that's completely wrong. The breakdown process actually begins before a single outside microbe even enters the picture.
You're referring to autolysis.
Autolysis. Self-digestion. Like the call is literally coming from inside the house.
It's a fascinating and honestly somewhat grim biochemical reality.
Break it down for me.
So while an animal is alive, its cells are full of highly destructive enzymes. Things like proteases, lipases, cathepsins.
What do those do?
They're essentially molecular scissors. In a living animal, they are very tightly controlled, compartmentalized within these little cellular structures called lysosomes.
Okay, so they're locked up.
Right. And they're used for normal cellular repair, breaking down old proteins, general metabolism, routine maintenance.
But when the animal dies, the control mechanisms fail. Like the security guards leave their posts.
Exactly. The cellular environment drastically changes. As we just discussed, oxygen ceases to circulate, glycogen is converted to lactic acid, and the internal pH drops.
Right.
This acidic environment actually degrades the membranes of those lysosomes. They rupture.
Oh, no.
Yeah. Spilling all these molecular scissors out directly into the muscle tissue. Suddenly, enzymes that were meant to maintain the muscle begin to aggressively dismantle it.
That is crazy. I picture it like a highly automated car factory, right?
Okay.
The managers, which are the living biological systems, they suddenly walk out, but the robotic arms on the assembly line are still powered up, still running their programming.
I like this analogy.
And without direction, they just start ripping the doors off the cars they just built, tearing up the conveyor belts, just completely dismantling the factory from the inside.
The factory analogy is really useful, but it's important to understand the specific targets of these robotic arms, these enzymes.
Okay, what are they targeting?
There are two primary avenues of destruction during autolysis. First is proteolytic action.
Proteo, meaning protein.
Right. The proteases begin cleaving the long, complex protein chains that make up the muscle fibers and connective tissue. This literally breaks down the structural integrity of the meat.
Which, ironically, is kind of what we want. To a degree, right? Like, that's what dry aging is.
Yeah, exactly. Controlled autolysis is tenderization. We use it to our advantage.
The enzymes are breaking down the tough connective tissue to make a steak tender.
Right, but uncontrolled excessive autolysis crosses a chemical line.
What happens then?
If it proceeds too far, The breakdown of these proteins releases amino acids and peptides that fundamentally alter the flavor profile. It leads to a defect that the industry calls souring.
Sour.
The meat physically deteriorates and develops this sour, really unpleasant tang purely from its own internal chemical collapse.
Right.
Completely independent of any bacteria.
Wow. So it can ruin itself before bacteria even show up.
Yes.
And what was the second avenue of destruction? You said there were two?
A slight hydrolysis of fats.
Oh, okay. Hydrolysis.
Lipase enzymes begin cleaving the fat molecules, the triglycerides, into free fatty acids.
And what does that do?
It subtly changes the flavor right off the bat. But more importantly, it makes the fats far more susceptible to further chemical degradation later on. It primes them for rancidity.
Okay, so the meat is basically softening his own tissues and breaking down his own fats, effectively preparing this like pre-digested banquet for the external bacteria when they finally arrive.
That is exactly what it's doing.
Which brings us to the invasion. The moment the microscopic hordes actually breach the perimeter, what controls the speed and severity of that initial invasion?
Well, this is where we have to look backwards, to the conditions surrounding the animal's death.
Okay.
The trajectory of spoilage is actually locked in based on several crucial pre-slaughter and slaughter factors.
You know, as a consumer, I never, ever think about this. I just look at the styrofoam tray at the supermarket and assume all chicken breasts or steaks started equal.
They absolutely do not start equal.
What's an example?
Consider the load in the gut of the animal.
The gut bacteria.
Right. An animal carrying a massive, highly active bacterial population in its digestive tract presents a severe risk during processing.
Because if there's an accident.
Exactly. Any minute cross-contamination during evisceration introduces an exponentially larger founding population of microbes directly onto the raw meat.
It's like more troops landing on the beach on day one.
Yes, but even more profound than that is the physical condition of the animal immediately prior to slaughter.
Really? How so?
Remember how we talked about glycogen acting as fuel and its conversion to lactic acid lowering the pH to 5.5?
Right, the acidic shield that kind of slows things down.
Now imagine an animal that is highly stressed, exhausted, or subjected to extreme physical exertion right before death.
Okay.
The fight-or-flight response dumps adrenaline into the system, and the animal actually burns through its muscle glycogen reserves while it's still alive.
Oh. Wow, I see the domino effect here. If the glycogen is burned up while the animal is alive,
There is no glycogen left after death to convert into lactic acid.
so the pH does not drop to that protective 5.5.
It does not. It stays abnormally high, maybe up around 6.5 or even a neutral 7.0.
Which is the microbe's absolute dream scenario.
Ah.
The natural acidic defense system is completely disabled from the start.
We call this DFD meat dark, firm, and dry.
Dark, firm, and dry.
Because the pH is so high, the proteins bind water incredibly tightly, making the meat look dark and feel dry to the touch. But microbiologically, it is highly, highly vulnerable.
It's a sitting duck.
It will spoil significantly faster than meat from a rested animal.
That is wild. The psychological and physical state of the animal literally dictates the molecular defense mechanisms of the resulting food.
It does. Biology is all interconnected.
What about the actual processing? Does that change things?
The method of killing and bleeding is paramount.
Feeding.
Blood is a nutrient-dense, highly oxygenated fluid. It is essentially a superhighway for bacterial growth.
Oh, gross.
If bleeding is incomplete and blood remains trapped in the vascular system of the muscle tissue, you are leaving behind deep internal reservoirs of perfect microbial food.
Oh, man. So they don't even have to work their way in from the outside.
Right. You allow spoilage organisms to penetrate deep into the meat rather than just colonizing the surface.
Okay, so the gut load, the pre-slaughter stress, the efficiency of bleeding, all of these factors predetermine the vulnerability of the meat.
Exactly.
But, by the time a family is standing in the grocery aisle looking at a package of pork chops, those factors are entirely out of their control. The die has been cast.
That's true.
So what is the very first variable that the consumer or even the grocery store actually controls?
The overarching dominant variable from the moment of slaughter straight through to your home refrigerator is the rate of cooling.
Temperature.
It is basic thermodynamics applied to biology. Microbes replicate enzymatically, and enzyme activity is highly, highly temperature dependent.
Okay.
Rapidly lowering the internal temperature of the carcass is the single most critical intervention we have. It slows down the autolytic enzymes tearing the meat apart from the inside, and it drastically increases the generation time of the invading bacteria.
Generation time, meaning how long it takes one bacterium to split into two.
Right. At room temperature, a bacterium might divide every 20 minutes.
20 minutes.
1 becomes 2, 2 become 4, 4 become 8. In just a few hours, you have millions.
Yikes.
But if you drop the temperature down to near freezing, that generation time might stretch to 12 hours or more.
So you are literally buying time.
Exactly. You're stretching the clock.
This is like the ultimate practical advice for anyone doing grocery runs.
Oh, absolutely.
If you buy a package of ground beef and then you leave it in your trunk on a warm afternoon while you stop at the hardware store and then you pick up dry cleaning, you aren't just letting the meat get a little warm.
No, you are drastically shortening generation times.
You are actively cultivating a bacterial colony in your Honda Civic.
You are accelerating the biological clock exponentially.
Yeah.
Every degree the temperature rises above freezing acts as an accelerant.
Wow. Okay, let's track this timeline forward. Autolysis has begun. The natural defenses are compromised. The temperature fluctuates a bit during transport. The bacterial invasion is underway.
The stage is set.
The type of damage these microbes inflict, the actual physical changes we see as consumers, depends almost entirely on one environmental factor, according to the sources.
Yes, oxygen.
Air.
Air is the great divider in the world of spoilage. It dictates entirely which microbial populations will dominate and what chemical weapons they will deploy.
Okay, so it splits into two paths.
We generally categorize spoilage into two distinct realms. Aerobic, meaning in the presence of oxygen, and anaerobic, meaning without it.
Let's start with the aerobic realm, because this is the scenario most people deal with daily, right?
Yes, very common.
The steak wrapped in that slightly permeable plastic film at the butcher counter, the leftover roast chicken sitting in a partially sealed Tupperware in the fridge. There is air circulating.
Right.
What is the very first physical manifestation that things have gone catastrophically wrong?
Under aerobic conditions, the most prominent and immediate visual and tactile cue is the development of surface slime.
Slime. Ugh. It is a deeply visceral, repulsive texture.
It definitely elicits a strong response.
When you touch a piece of raw poultry and it feels slick or sticky or viscous, your brain immediately screams, danger.
And it should.
What exactly is that slime on a microscopic level? Because it's not just meat juice.
No, it is not simply moisture or fat breaking down. That slime is a highly structured, dense metropolis of living bacterial cells.
A metropolis.
In aerobic environments, specifically on the surface of meat where oxygen is readily available, we see an explosion of specific bacterial genera, things like Pseudomonas moraxella, Streptococcus, Bacillus, and Micrococcus.
Why do these specific bacteria produce slime, though? Is it just a byproduct of their digestion, like bacterial sweat?
It's far more insidious and coordinated than that.
Coordinated?
Yes. As these bacteria multiply on the surface, they don't just exist as isolated individuals bumping into each other. They engage in a process called quorum sensing.
Quorum sensing. Like taking a vote.
Exactly. When their population density reaches a critical threshold, they can essentially sense each other, and they trigger a collective genetic response.
Wow.
They all simultaneously begin secreting massive amounts of extracellular polymeric substances, or EPS.
Extracellular polymeric substances. Okay, that sounds like a mouthful. What is it actually made of?
Essentially complex sugars and proteins that form protective gelatinous matrix.
Slime.
Yes, this is a biofilm. The slime you feel is this biofilm.
Why do they build it?
They build it to protect themselves from environmental stress, to trap nutrients, and crucially, to lock moisture against the meat's surface so they don't dry out.
Oh my God. So when you feel slime, you are literally running your finger over the architecture of a microscopic city containing billions of bacterial cells.
That is exactly what you're doing.
That is horrifying. I will never touch a slimy chicken breast the same way again.
That's a good survival instinct.
But this brings up a massive point about how we behave in our kitchens. So many people treat the best by or sell by date on the package as an absolute magical force field.
Which is very dangerous.
Right. If the sticker says it's good until Friday, people will blindly cook it on Thursday, even if it feels like it's coated in mucus.
That is a critical failure in food safety logic. The formation of surface slime is an early indicator of exponential microbial growth.
Meaning it's already bad?
Yes. It frequently occurs well before the printed expiry date, especially if the temperature was poorly managed during transit or in the consumer's home.
So the date on the package is basically a theoretical projection.
Entirely theoretical.
It's based on perfect, unbroken cold chain logistics. It assumes the meat stayed exactly at 34 degrees Fahrenheit from the slaughterhouse directly to your frying pan.
Which almost never happens.
Right. If the meat sat on a loading dock in the sun for an hour, or if your home fridge is running slightly warm, Pseudomonas does not care what the ink on the sticker says.
Exactly. The biological reality supersedes the printed date. Every time. If you detect slime, the population of Pseudomonas and its allies has already reached critical mass.
This city has been built.
The meat is actively spoiling. Do not eat it.
Okay, so slime is just the tactile warning. But aerobic bacteria also wage a highly visible chemical war on the actual color of the meat.
They do, and it's fascinating chemistry.
We buy beef specifically because of that beautiful, bright cherry red color, right? It looks fresh, it looks healthy.
The industry calls it the bloom.
The bloom.
The bloom is highly prized by consumers, but it is chemically incredibly fragile. To understand why bacteria destroy it, you have to understand what the color actually is in the first place.
Okay, lay it on me.
That red color comes from a protein called myoglobin, which is responsible for storing oxygen in muscle tissue.
It's similar to hemoglobin in our blood, right? It has an iron atom sitting in the middle of it.
Correct. It contains a porphyrin ring with an iron atom right at its center. When meat is freshly cut and exposed to the air, oxygen binds to that iron atom.
And that creates the color.
It creates oxymyoglobin, which is that brilliant cherry red color.
So the air itself makes it look fresh initially.
Yes, initially. But the very oxygen that creates the red color is also fueling the aerobic bacteria on the surface.
Ah, double-edged sword.
And as specific bacteria, particularly species of lactobacillus, proliferate, they produce powerful oxidizing agents as metabolic waste.
Oxidizing agents. Remind me, those are chemicals that strip electrons away from other molecules.
Precisely. Lactobacillus pumps out compounds like hydrogen peroxide and hydrogen sulfide.
Hydrogen peroxide, like the stuff in the brown bottle in my bathroom?
The exact same chemical. And these chemicals interact directly with the oxymyoglobin. The hydrogen peroxide oxidizes the iron atom in the myoglobin, changing its valence state from Fe2+ to Fe3+ cell.
Okay, we're getting deep into the chemistry now. F2+ to F3+ have. What does that valence change actually look like to the human eye? Like, what happens to the steak?
It changes the entire molecular structure of the pigment, converting the bright red oxymyoglobin into a completely different compound called metmyoglobin.
Myoglobin.
And metmyoglobin is a dull, unappetizing, grayish-brown color.
So when I pull a steak out of the fridge and it has turned gray or brown, it's not just drying out.
No, it's a chemical reaction.
I am looking at a literal chemical bleaching event. The lactobacillus has manufactured hydrogen peroxide right on the surface of the meat and chemically oxidized the iron in the muscle tissue.
It is an active, microbially mediated chemical degradation.
That is insane.
And unfortunately, the color changes can get much more extreme and bizarre than just gray or brown.
Wait, worse than gray?
Oh, yes. Under certain aerobic conditions, you can encounter a terrifying spectrum of pigmented bacterial colonies.
The rainbow of disgust.
That's a very apt description.
What colors are we talking about?
Well, if you observe vivid red spots developing on the surface, you are likely looking at colonies of Serratia marcescens.
Red spots on meat. But meat is already red.
It's a very distinct bright red, almost like a dye. This bacterium produces a red pigment called prodigiosin as a secondary metabolite.
So it's actually synthesizing red dye on my chicken.
Essentially, yes.
What else?
If you see peculiar blue patches, the culprit is often Pseudomonas cincinnea, which secretes complex blue pigmented molecules.
Blue meat. Unbelievable.
And if you encounter greenish blue or brownish black spots, you are dealing with colonies of organisms like Chromobacterium lividum.
Okay, I cannot emphasize this enough. If any parent or home cook sees a blue or green spot on their raw chicken, I don't think they need a degree in microbiology to throw it out.
I would hope not.
It is astonishing, though, that these bacteria have such specific, vibrant color signatures.
It definitely serves as an undeniable visual alarm for us. But while the proteins and pigments are under attack, the fat is also being degraded.
Okay, let's talk about the fat.
However, the breakdown of fat in an aerobic environment is primarily a chemical reaction, though microbes can absolutely accelerate it.
We're talking about fat oxidation, right? Rancidity.
Yes, oxidative rancidity. When the unsaturated fatty acids in meat are exposed to oxygen, they undergo an autooxidation chain reaction.
Autooxidation.
Oxygen molecules attack the double bonds in the fat molecules, creating free radicals.
Free radicals, those are those highly reactive, unstable molecules that just like run around smashing into other molecules, stealing their electrons and creating more free radicals, right?
It's exactly that, a cascading chemical collapse. These free radicals break down the complex fats into smaller, highly volatile compounds, specifically aldehydes and ketones.
And what do aldehydes and ketones taste like?
They possess very harsh, cardboard-like, rancid odors and flavors. It's that classic old fat smell.
Gross.
Now, aerobic bacteria like Pseudomonas or certain yeasts can produce enzymes that initiate this process, but a crucial detail is that this oxidation is powerfully catalyzed by non-biological environmental factors.
Catalyzed meaning sped up.
Drastically sped up.
Why what?
The initiation of this free radical cascade is accelerated by exposure to ultraviolet light and, incredibly, by trace amounts of transition metals.
Transition metals.
Specifically, copper.
Copper. Okay, this blew my mind when I read the sources.
It's a really interesting quirk of chemistry.
So if someone has a trendy, beautiful copper countertop in their kitchen, right?
They're very popular right now.
And it's situated right under a bright sunlit window. If they take a piece of raw meat and leave it sitting on that counter to come up to room temperature before cooking,
they are essentially constructing the perfect chemical reactor for destroying the fats in that meat.
that is so specific and crazy.
The ambient UV light from the window provides the activation energy, and the copper ions on the counter act as a powerful catalyst. They rapidly transfer electrons and initiate that free radical chain reaction in the lipids.
So the fat just breaks down instantly.
The fat will become oxidized and rancid far quicker than it would on a stainless steel surface in the dark.
That is the most specific, incredible food chemistry warning I've ever heard. Listeners, keep your steaks away from sunlight and copper countertops.
It's very sound advice.
Okay, so in the aerobic realm, we have slime cities built by Pseudomonas. We have hydrogen peroxide bleaching the red myoglobin into gray metmyoglobin. We have Serratia painting the meat red, and copper-catalyzed fat fires.
A busy ecosystem.
But bacteria aren't the only microbes that thrive in oxygen, right?
They are not.
Yeah.
We must address the fungi.
Oh, boy.
The aerobic realm is also the domain of molds.
The fuzz. The absolute bane of every Tupperware container shoved to the back of the fridge and forgotten about for 3 weeks.
We all have one in there somewhere. Molds are remarkably resilient aerobic organisms, and they cause highly specific physical and chemical alterations.
Like what?
Like bacteria, they can contribute to surface stickiness. They also produce characteristic pigmented patches. For example, the classic powdery green patches you often see on old food are typically species of penicillium.
Wait, penicillium, the exact same genus that gives us penicillin antibiotics.
The very same.
Wow.
Although in your refrigerator, they aren't saving lives. They are just opportunistic scavengers decomposing your leftovers.
Good to know I can't cure strep throat by eating moldy chicken.
Please do not try that.
Noted. But, okay, the most distinct visual manifestation of mold on refrigerated meat isn't just green patches, right? It's the development of what the sources call whiskers.
Whiskers, yes.
Whiskers. It sounds almost benign, kind of cute, but I know it's actually a structural invasion.
It is the fungal architecture taking over.
Explain the whiskers to me.
A mold organism essentially consists of two parts. There are the spores, which are the reproductive seeds that float in the air and land on the food.
Right.
Once a spore germinates, it begins growing a vast branching network of thread-like roots called mycelium.
So the fuzz we see on the outside isn't the whole organism, it's just the root system reaching out into the air.
Exactly. Normally, a mold will grow its mycelium and then develop specialized structures to release more spores, which is called sporulation.
Okay, so it grows, then it reproduces.
Right. However, when meat is stored at temperatures near freezing, which is the environment of a standard home refrigerator, the cold temperature actually inhibits that final reproductive stage.
Oh.
The mold cannot sporulate.
But it can still grow the roots.
Yes. You see a limited but very persistent amount of mycelial growth without sporulation.
The whiskers.
These white, fluffy, hair-like projections extending out from the surface of the meat are the whiskers.
I want to hammer this point home for everyone listening, especially parents trying to stretch their grocery budget or manage weekly meal prep. This completely obliterates the pause button myth of the refrigerator.
It really does. Elaborate on the myth for a second.
Okay, so we intuitively believe that the cold temperature of a fridge puts food in, like, biological stasis, right?
Right. A lot of people think that.
We think that hovering right around 35 degrees Fahrenheit means life just stops. We think that if we put leftover roast beef in a sealed container, we literally freeze time.
But the Whiskers prove that time and biology march on.
They don't care about 35 degrees.
Why do molds succeed in the cold when many bacteria fail, though?
That's a great question. Why?
It comes down to water activity and competitive exclusion.
Oh, back to water activity.
As meat sits in the fridge, the surface slowly dries out. Cold air is very dry air.
Right.
Now, bacteria require a very high water activity to swim, to transport nutrients, and to divide. As the surface dries, bacterial growth slows dramatically.
But molds don't need a swimming pool.
Precisely. Molds are adapted to thrive in environments with much lower water activity.
So the cold slows down the bacteria.
And with the bacteria slowed down by the cold and the drying surface, the molds suddenly face much less competition for nutrients.
They have the whole buffet to themselves.
They do, and they slowly, relentlessly extend their mycelium.
So the fridge is merely a delay tactic. It suppresses the bacterial slime, sure, but it hands the keys to the kingdom over to the fungal whiskers.
That's a perfect way to summarize it. And those whiskers are not just an aesthetic issue, by the way.
They are.
While they grow, they are excreting enzymes directly into the meat. Many of these aerobic molds possess powerful lipase enzymes.
Like K's, like the autolytic enzymes we talked about earlier, enzymes that destroy lipids or fats.
Yes, the mold secretes lipase, which actively causes the hydrolysis of the meat's fat stores.
Breaking down the fat.
It chemically cleaves the triglyceride molecules apart, breaking them down into free fatty acids and glycerol.
And what does that do to the taste?
This profound chemical decomposition leads to intensely soapy, rancid, and completely undesirable flavors. The mold is literally digesting the fat from the outside in.
Wow. Okay, so let's look at the problem logically.
Right.
If oxygen is the ultimate enemy-like, if air is what enables the Pseudomonas slime, the lactobacillus gray bleaching, the copper-catalyzed rancidity, and the fungal whiskers.
Yes, oxygen drives all of that.
The solution for the modern food industry seems super obvious, right? Just eliminate the air, suck it all out, vacuum packaging.
It is the logical engineering solution, and it's widely used. Vacuum sealing, or modifying the atmosphere by replacing oxygen with nitrogen or carbon dioxide, it completely shuts down those aerobic spoilage pathways.
Because they can't breathe.
Pseudomonas cannot breathe. Penicillium cannot grow its whiskers. The red bloom is altered, sure, but the massive rapid aerobic decay is halted.
But nature abhors a vacuum, right? Removing the oxygen doesn't make the meat immortal. It just shifts the ecological balance of power.
It introduces us to the vacuum realm. Welcome to anaerobic spoilage.
The domain of microbes that don't need air.
Specifically, facultative anaerobes and strict anaerobes.
What's the difference?
Facultative means they can survive with or without oxygen. They simply shift their metabolic pathways depending on the environment.
Okay, they're flexible.
Strict anaerobes, however, are poisoned by oxygen. They can only thrive in a true vacuum.
And the type of destruction they cause is fundamentally different, isn't it? Like, aerobic spoilage is highly visual, slime, fuzz, rainbow spots. Anaerobic spoilage seems much stealthier.
It is deeply chemical and intensely olfactory.
It's all about the smell.
In a vacuum-sealed package, you will almost always smell the spoilage long before you see any structural breakdown.
Okay, so what's the first sign?
The first major phase of anaerobic spoilage is generally characterized by souring.
Now wait, we mentioned souring earlier with autolysis, the meat's own enzymes breaking it down. Is this the exact same chemical process?
It results in a very similar sensory profile, sour tang.
Yeah.
But the mechanism is entirely microbially driven.
Okay, how does that work?
In the absence of oxygen, bacteria cannot utilize normal cellular respiration. They must rely exclusively on fermentation to generate energy.
And when you ferment sugars and proteins without oxygen, you get acids.
Massive amounts of them. Facultative bacteria, particularly species of lactic acid bacteria, rapidly ferment the remaining carbohydrates and produce a heavy load of volatile organic acids as waste.
What kind of acids?
The primary culprits are formic acid, propionic acid, and acetic acid.
Acetic acid, that is the primary component of vinegar, right?
Exactly. So these bacteria are essentially pickling the meat from the inside out with their waste products.
That sounds crazy.
When you open a vacuum-sealed package that has undergone this souring, you are hit with a sharp, tangy, almost vinegar-like aroma.
Okay, that sounds unpleasant, but not necessarily dangerous. I mean, people eat fermented sausages all the time.
That's true. Controlled fermentation is safe.
But souring is just the preamble, right? if the conditions are right, the vacuum realm descends into something much darker.
It descends into putrefaction.
Putrefaction. The word itself just sounds heavy and foul.
It is a very specific biochemical term. Putrefaction is strictly defined as the anaerobic decomposition of protein.
So the protein itself is rotting.
It is the breakdown of the very structural foundation of the meat without the presence of oxygen. And the primary architects of putrefaction are bacteria belonging to the genus Clostridium.
Clostridium. Okay, these are the heavy hitters of the microscopic world, like Clostridium botulinum causes botulism, Clostridium tetani causes tetanus.
They are not to be trifled with. They are formidable, strict anaerobes.
What do they do to the meat?
They possess enzymes capable of entirely dismantling complex protein molecules, specifically attacking the amino acids.
How?
Through a process called decarboxylation, Clostridium strips the carboxyl group off amino acids, creating incredibly potent, toxic, and foul-smelling compounds known as biogenic amines.
Biogenic amines. What do these actually smell like? Give me the chemical profile of putrefaction.
It is a cocktail of deeply offensive odors.
Lay it on me.
They produce hydrogen sulfide,
Okay.
which provides the distinct choking smell of rotten eggs.
Okay, rotten eggs. Got it.
They produce indole, a compound that at high concentrations possesses an intensely fecal odor.
Oh, God.
They generate ammonia, which is sharp and caustic.
Right.
And they produce specific biogenic amines with incredibly descriptive names: putrescine and cadaverine.
Putrescine and cadaverine, the literal smells of rotting flesh and cadavers.
Yes. These compounds are the chemical signature of death and severe anaerobic decay.
That is just so incredibly visceral.
When Clostridium is active in a vacuum package, it is dismantling the muscle tissue and converting it into a slurry of ammonia, rotten eggs, and cadaverine.
Which is an unmistakable biological warning that the material is highly toxic.
Exactly. Your body reacts with revulsion for a reason.
Okay, so the smell is the ultimate alarm in the vacuum realm, but there is one visual cue associated with anaerobic spoilage in vacuum packaging that is incredibly common and honestly a bit terrifying.
I know exactly what you're talking about.
Fast production.
The ballooning package.
Yes. We've all seen it. You're at the grocery store, maybe digging through the discounted meat bin, or you check a package you've kept in the fridge way too long.
Right.
The heavy plastic film, which is supposed to be vacuum-pulled skin tight against the beef, is puffed outward.
It looks like an inflated pillow.
It looks like it's going to pop. It's so taut.
That physical inflation is the direct result of intense microbial respiration and fermentation occurring in a sealed environment.
Because the gas has nowhere to go.
The bacteria are consuming nutrients and excreting gas molecules, which can't escape the plastic. The pressure builds, inflating the package.
But here is where I need you to clarify a massive nuance for the consumer, because it's not always black and white, is it?
No, it's not. You're referring to the difference between Clostridium gas and lactic acid bacteria gas.
Exactly. Because if my vacuum-sealed beef is puffed up, it could be Clostridium.
It certainly could.
Which means they are actively engaging in putrefaction, making putrescine and hydrogen sulfide. If I puncture that package, my kitchen is going to smell like a literal sewer, and the meat is a toxic biohazard.
Yes, Clostridium species are prolific gas producers alongside their putrefactive activities.
But on the other hand, it could just be lactic acid bacteria.
Correct.
The sources mention that certain heterofermentative lactic acid bacteria produce carbon dioxide gas as a byproduct of their fermentation without engaging in the extreme protein breakdown that causes putrefaction.
That's right.
So the package puffs up with CO2, but it might not smell like literal death. It might just smell a bit sour.
Exactly.
So how is a consumer supposed to know what kind of gas is stretching that plastic?
That is the insidious nature of vacuum packaging. From the outside, the visual cue is completely identical.
You can't tell the difference by looking.
A balloon package simply tells you that extreme microbial metabolism is occurring. The population has exploded. The biological clock has run out.
So even if it's just CO2 from lactic acid bacteria, the fact that it has produced enough gas to inflate that heavy plastic means the bacterial load is astronomically high.
Precisely. To definitively know the chemical makeup of the spoilage, you would have to rely on your olfactory senses.
You'd have to open it and smell it.
You'd have to open the package and smell the difference between sour acetic acid and foul putrescine.
Which sounds like a terrible game to play in your kitchen.
It is. So, as a consumer safety rule, the source of the gas is irrelevant.
Irrelevant.
The physical presence of severe ballooning indicates that the microbial load has surpassed any acceptable safety threshold. A puffed package is a compromise package. period.
Don't sniff the balloon, just throw it away.
Exactly.
This highlights a crucial theme for this whole episode. Whether we are dealing with the oxygen-rich environment of a butcher's display case or the oxygen-starved vacuum of an industrial plastic seal, the ultimate test for the consumer relies on decoding these final volatile chemical alarms.
We really do.
We have to train ourselves to read the chemistry of odors and colors.
It requires treating our senses like chemical spectrometers.
I love that phrase.
The microbial world leaves highly specific signatures. Let's examine the olfactory signatures of aerobic spoilage first, because they are surprisingly complex and often very deceptive.
Deceptive is the right word, because look, if I smell ammonia or rotten eggs, I know it's bad. My brain says no.
Instinct takes over.
But there are off odors associated with aerobically stored meat that don't immediately scream danger. For example, sweet or fruity odors.
Yes, the sweet and fruity phase. This is an incredibly common trap for consumers.
Walk me through it.
They open a container of chicken, give it a smell, and instead of a neutral meat scent, they detect something faintly floral, sweet, or almost reminiscent of overripe fruit.
And the brain thinks, well, fruit isn't terrible. Maybe it was a marinade I used. Maybe it's fine.
It is not fine. That fruity aroma is a biological trick, a byproduct of the early growth phases of Pseudomonas bacteria.
Wait, Pseudomonas? The slime guys?
The very same. As Pseudomonas begins breaking down the amino acids on the surface of the meat, they utilize specific enzymatic pathways that produce volatile organic compounds called esters.
Esters.
Yes.
Wait, aren't esters the exact same class of chemical compounds that give actual fruits like pineapples and bananas their smell?
Precisely. The bacteria are literally manufacturing the chemical equivalents of fruit scents.
That is devious.
But in the context of raw meat, the presence of these esters is definitive proof of advanced bacterial colonization. The sweet smell is the herald of the slime that is soon to follow.
So if your chicken smells like a faint fruit salad, throw it out. It's Pseudomonas throwing a party.
A party you don't want to attend.
What about other aerobic odors?
As the aerobic breakdown continues, the metabolism of sulfur-containing amino acids by various bacteria leads to the release of complex sulfur compounds.
Okay, more sulfur.
But these don't necessarily smell like the sharp hydrogen sulfide of anaerobic putrefaction. Instead, they can manifest as intensely cheesy odors.
Cheesy on raw meat.
Yes. And again, people might assume that, much like dry aging, the meat is just developing a complex aged profile.
Like it's getting fancy.
Right. But unless it was strictly controlled under precise humidity and temperature parameters in a specialized facility, a cheesy smell on raw poultry or pork is not culinary perfection.
What is it then?
It is the volatile sulfur compounds of uncontrolled bacterial decay.
Sweet, fruity, cheesy. We really do have to completely unlearn our positive associations with these smells when it comes to raw meat.
Context is everything in chemistry.
Absolutely. But while the nose detects the volatile compounds in the air, the eyes must detect the chemical alterations on the surface.
Yes, visual cues are just as important.
We touched on the loss of the red bloom, the myoglobin turning into gray myoglobin. But there is one specific color change that is so dramatic, so chemically fascinating, that we have to break it down.
I know where you're going with this.
Green meat.
Ah, yes. The formation of green pigments is a remarkable intersection of bacterial metabolism and muscle chemistry.
And just to be clear, it is completely distinct from the green fuzz of penicillium mold, right?
Yes, completely different. This is the meat tissue itself turning a vivid, iridescent green.
Walk us through the biochemical pathway of green meat, because it's not just a stain.
No, it begins with specific strains of bacteria, often species of Lactobacillus or Leuconostoc, thriving on the surface.
Okay.
As they metabolize, their specific waste product is hydrogen sulfide gas, H2S.
The rotten egg gas again?
Yes, but in this aerobic environment, the gas doesn't just dissipate into the air. It reacts directly and violently with the myoglobin pigment in the muscle tissue.
Oh, it attacks the protein.
The hydrogen sulfide attacks the porphyrin ring of the myoglobin molecule.
The structure holding the iron atom.
Exactly. The chemical reaction structurally alters the myoglobin, converting it into a completely new, permanently modified compound called sulfmyoglobin.
Sulfmyoglobin?
And sulfmyoglobin possesses a distinct, bright green color.
So when I see a green sheen on a slice of roast beef or a pork chop, I'm not just looking at a surface stain.
No, you're not.
I am looking at a localized chemical factory. The bacteria have pumped out enough toxic hydrogen sulfide gas to fundamentally chemically alter the molecular structure of the meat's native proteins.
It is a permanent chemical scar indicating severe bacterial contamination. You cannot cook it out.
Wow.
You cannot wash it off. The protein itself has been irrevocably changed by microbial waste.
It is incredibly helpful to understand the why behind the green rather than just knowing green is bad.
It demystifies the process.
And what becomes abundantly clear across this whole deep dive is how specific environments curate specific microbial threats.
Absolutely. The environment is the selector.
Like the oxygen-rich surface curates the Pseudomonas slime and the Sulfmyoglobin green.
Right.
The oxygen-starved vacuum curates the Clostridium Putrefaction and the lactic acid souring. The environment dictates the dominant predator.
And we see this environmental curation even within specific types of meat products. Processed meats, for example, possess an entirely different ecological landscape than fresh cuts.
Processed meats, like hot dogs, deli slices, bologna, sausages.
Yes. These products have been heavily modified by humans.
How so?
They've been cooked, altering the protein structures. They have high concentrations of salt added, which drastically lowers the water activity.
Right, making it harder for bacteria to thrive.
And they often contain chemical preservatives like nitrates.
Which are added specifically to suppress Clostridium botulinum, right?
Exactly. These modifications make processed meats incredibly hostile to the typical fresh meat spoilers like Pseudomonas.
So they should be safe forever.
But nature always finds a way.
Of course it does.
The harsh chemical environment of processed meat creates a hyper-specific niche that selects for highly specialized, incredibly resilient bacteria.
Who are the champions of the hot dog package?
Primarily species of Microbacterium and Extremophile.
Never heard of them.
These organisms have evolved extremely high salt tolerance and resistance to common preservatives.
So they just don't care about the salt or the nitrates.
They brush it off. When you see a slimy film developing on deli ham, or a milky, cloudy liquid pooling in a package of hot dogs,
Oh, the hot dog water.
you are witnessing the handiwork of these highly specialized extremophiles. They conquer the environments that destroy other bacteria.
It really is an arms race. We invent vacuum sealing and Clostridium steps up.
Yes.
We invent nitrate curing and Microbacterium steps up.
Yeah.
We invent industrial refrigeration, holding entire warehouses of beef at exactly 0 degrees Celsius, right at the freezing point of water.
And even there, at the absolute freezing point, a challenger emerges.
The final boss of the cold storage world, who is it?
We discuss how home refrigerators, operating slightly above freezing, allow molds to grow whiskers.
Right.
But in prolonged industrial cold storage, where massive beef sides or quarters are held strictly at 0 degrees C for weeks or months, a very specific fungus becomes the dominant threat.
What's its name?
Cladosporium.
Cladosporium.
This specific mold has adapted its cellular machinery to remain metabolically active even when the surrounding water is on the absolute precipice of freezing solid.
How is that even possible?
Its enzymes continue to function, its mycelium continues to spread, causing distinct black spot spoilage on the surface of the beef.
Black spots at 0 degrees.
It proves that there is virtually no temperature or atmospheric condition that can permanently halt biological decay. You simply alter which specific microbe has the home field advantage.
It's humbling, honestly.
The microbial world is very patient.
So after this exhaustive journey through the microscopic war zone, let's synthesize the ultimate practical consumer lesson.
Let's do it.
We've covered the chemistry, the physics, and the biology. If you, our listener, are standing in front of your open refrigerator tonight, staring down a piece of meat, what are the absolute non-negotiable, chemically backed signs that it is unsafe to eat?
The red flags are unambiguous and rooted entirely in the mechanisms we've discussed today.
Okay, number one.
Number one, the tactile test. Any presence of surface slime or stickiness.
The slime city.
This is not moisture. This is the physical biofilm of an aerobic bacterial colony, likely Pseudomonas. Its presence immediately invalidates any printed expiration date.
Throw it out. Number two.
Number two, the visual test. Look closely at the color.
We want the red bloom, but what are we looking out for?
Any loss of the healthy red oxymyoglobin bloom to a dull gray or brown myoglobin indicate active chemical oxidation by bacterial waste like hydrogen peroxide.
Okay, gray or brown is bad.
Furthermore, any vivid discolorations, The red of Serratia, the blue of Pseudomonas cincinnati, or crucially the iridescent green of Sulfmyoglobin, indicates severe, localized chemical alteration by microbial toxins.
Yeah. If it looks like a rainbow, throw it out. Number 3.
Number 3, the olfactory test. You have to trust your nose, but you have to know what you are smelling for.
We're detecting volatile organic compounds.
Yes. Be deeply suspicious of sweet, fruity ester smells, which are early Pseudomonas warnings.
No fruit salad chicken.
Cheesy sulfur odors are alarms. Sharp, vinegar-like sourness indicates heavy acid production.
Right.
And obviously, any deeply foul smells of ammonia, rotten eggs, or fecal notes indicate the release of biogenic amines and active putrefaction.
Cadaverine and putrescine?
If it does not smell strictly like neutral, fresh tissue, the microbes have achieved dominance.
And finally, number 4, the physical geometry of the packaging.
Any ballooning, puffing, or gas accumulation within a vacuum-sealed or modified atmosphere package.
The balloon.
The physical expansion of the plastic is tangible evidence of massive microbial respiration and fermentation. The biological load is too high to risk consumption.
If you see, smell, or feel any of these signs, you aren't just risking a bad-tasting meal. You are ingesting an accumulation of microbial waste acids, enzymes, amines, and potentially toxins that your body will violently reject.
The food is no longer food. It is a thriving, toxic ecosystem.
Well, this has been an incredibly illuminating, mathematically precise, and admittedly somewhat stomach-churning journey.
I hope it wasn't too gross.
Oh, it was exactly the right amount of gross. We want to thank you, our listener, for braving this deep dive with us today.
Yes, thank you for sticking with it.
We've traced the timeline of a piece of meat from the physical and emotional stress of the animal's final moments, right through the internal self-destruction of lysosomal autolysis.
We covered a lot of ground.
We've mapped the aerobic slime cities, tracked the chemical bleaching of myoglobin, marveled at the near-freezing fungal whiskers, and descended into the anaerobic, gas-producing depths of putrescine and cadaverine. I guarantee you will never look at grocery shopping the exact same way again.
It requires a paradigm shift in how we view our kitchens, honestly.
It really does.
But there is one final, perhaps slightly unnerving consequence to all of this that we haven't even touched upon yet.
Oh boy, what is it?
We've discussed how bacteria adapt to different environments. Salt, vacuums, cold. But think about the environment of your own refrigerator over the span of years.
Over years? What do you mean?
Well, we constantly cycle raw meat in and out of our fridges. Every week, we introduce Pseudomonas, lactobacillus, and molds. Yeah. And then what do? We clean our fridges, we use chemical sprays, we wipe them down with bleach, we lower the temperature. We think we are sanitizing the environment.
Are we not?
We are actually just creating an evolutionary proving ground.
Oh, wait, what?
Exactly. By constantly introducing meat bacteria into the cold, isolated environment of the kitchen fridge and then bombarding them with chemical cleaners, we are subjecting them to extreme selective pressure.
Survival of the fittest in my CRISPR drawer.
Yes. Through horizontal gene transfer, which is where bacteria literally swap pieces of DNA with each other, the microbes that manage to survive in the microscopic scratches of your plastic shelves are constantly sharing resistance traits.
Oh my God.
They are sharing cold tolerance genes, they're sharing biofilm production genes, resistance to your specific kitchen cleaner.
So over time, the microbiome of our own kitchens isn't just a random collection of germs. We are actively domesticating and evolving bespoke, hyper-resilient strains of spoilage bacteria that are perfectly adapted to defeat our specific home storage methods.
It is a microscopic arms race happening right next to your milk.
That is terrifying.
We build better refrigerators. The microbial world simply builds a better Pseudomonas. The war is never won. The battle lines just constantly shift.
Wow.
And the next time you open that door, remember,
Yeah.
You're not just looking at a storage unit. You're looking at a highly evolved, heavily contested biological battlefield.